So welcome everybody. Thank you for coming today to this week's meeting of the IMAG MSM Working Group on Multi-Scale Modeling and Virtual and Viral Pandemics. Just a reminder that today's meeting, like all other meetings, is being recorded and live streamed, and will be available through our YouTube channel after the fact for future viewing. Just a few announcements to go over before we get started, and then we'll turn things over to Paul Roberts, our speaker today, from the University of, is it Birmingham or Birmingham?
Birmingham.
Birmingham. Yeah, because we would say Birmingham here in the
Sure.
States. So I wanted to pronounce it correctly. But all right, so just a quick reminder on leadership for the group. So this is co-led by Tom Helikar and James Glazier, with web administration by Jim Sluka, and activity coordination by Lorenzo Vescini. And if anybody has any questions, comments, or requests for any of them, their information can be found here. And these slides also will be available through the working group wiki page once this presentation is concluded. Slides will usually go up by tomorrow. Some links to our various social media groups. We have a Slack channel for various conversation and guest scheduling that everybody's welcome to join. You can connect with us on Twitter. Announcements are listed through the IMAG MSN wiki page, as previously mentioned. We have a LinkedIn page as well with links here, and our YouTube channel with this talk and all previous talks available for viewing. Again, these slides will be available for download if you need reference of these links later. Just a reminder of the ongoing Glimprint initiative that is currently being led by Tom Helikar, I believe. If anybody is interested in getting involved in the Glimprint, the development of the Glimprint website and group, please feel free to contact using the links on the Glimprint website. Again, please follow us on the Twitter group, now called X, I suppose. If you could retweet weekly announcements, there's usually a couple for each speaker. That would be great to help boost attendance and community awareness. One last announcement, we do have a faculty position currently being advertised at the University of Florida in computational systems medicine. Anybody who's interested in applying for this or getting more information, maybe for their graduating students, they can contact Dr. Reinhard Launbacher at the email address listed here. Sorry, a couple more announcements. We do have our CompuCell 3D training workshop coming up at the end of next month. Anybody who's interested in attending, or again, who has students who may be interested in attending, you can find more information on our website listed below, or you can contact me. I'm the coordinator for that this year. Another workshop leading up to that on focusing more on network modeling in Python, led by Dr. Herbert Sauro at University of Washington, will take place a week before ours. And you can register using the links on this page. I think the deadline for that is actually, registration deadline for that is tomorrow. So if you or anyone you know might be interested in that, you might want to go ahead and get registered as quickly as possible. If you have any publications to share with the group, please feel free to submit them through the wiki page. You can email Jim Sluka to get those posted there to kind of broadcast your recent publications. And I think with that, so we can open the floor. I think anybody else has any brief announcements. One last thing, there will be no steering committee tomorrow at 11, like there usually is, because I think half of the leadership is currently out of town at conferences or on vacation. So any other announcements about meetings, funding, or requests for help or anything like that? Okay, I think that's probably a no. We can circle back around later if something does come up. But with that, I think I'll go ahead and turn things over. Sorry, one last slide. Just a quick reminder of upcoming meetings. So July 11th, currently still looking for a presenter. If anybody has speakers who they'd like to suggest, please feel free to use our Google form for submitting that. July 25th, we'll have Francesco Pappalardo from University of Canania. And then on August 8th, we'll have Bishop Paldella from University of Virginia. But again, if you have any speakers you'd like to suggest or know anybody who may have suggested speakers, please feel free to get in touch with us. All right, and with that, I will go ahead and turn things over to Paul Roberts from the University of Birmingham, who will be talking to us today about mathematical models to elucidate mechanisms underlying sight loss.
Thank you ever so much for the kind invitation to speak. As Hayden was saying, my name is Paul Roberts. I'm currently a Centre Fellow at the Centre for Systems Modelling and Quantitative Biomedicine at the University of Birmingham in the UK. Although I should note that from the 15th of July of this year, I'll be taking up a post as a lecturer in the Department of Optometry and Visual Sciences at City University of London. So if you're listening to the recording in a few weeks' time, I'm there. So I'd like to talk to you today about mathematical models to elucidate the mechanisms underlying sight loss. And in particular, I'm looking at a group of inherited conditions which are known collectively as retinitis pigmentosa, or RP. And I'll tell you a bit more about those in a moment. So just to say, in terms of acknowledgement, so this work that I'm going to share with you today was actually completed at the University of Sussex, during which time I was funded by BBSRC. It was completed alone, so there were no collaborators to list. So I'd like to begin by giving a couple of definitions. So I work in a field for which I have coined the term, sorry, I'm trouble with this slide, for which I've coined the term mathematical ophthalmology. So I define this term as using mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease. So using mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease. And this term runs parallel to an existing term, which is computational ophthalmology, which I define as using computational algorithms to preserve vision by identifying and predicting ocular features and risks. So using computational algorithms to preserve vision by identifying and predicting ocular features and risks. So as far as mathematical ophthalmology goes, this is a mechanistic approach to modelling. So it would encompass both sort of a mathematical biology or computational biology type approach. Whereas in terms of the way that computational ophthalmology has been defined, that would essentially be artificial intelligence type approaches. So in each case, there are three ways in which this is helpful. Firstly, they improve our understanding, enable us to gain a deeper understanding than we would get through purely experimental or clinical studies. They enable us to make predictions about how a system will behave in healthy, diseased, or various perturbed states. And they also enable us to develop more effective treatments or eventually to develop personalized treatment strategies. They each have their strengths and weaknesses. They're not at all in competition. They're very much complementary approaches. As you would imagine from my description, mathematical ophthalmology has the advantage of giving a high degree of mechanistic insight, whereas computational ophthalmology would give low or generally no mechanistic insight because the artificial intelligence algorithms don't themselves contain a description of the underlying mechanisms. On the other hand, AI type approaches can be incredibly powerful predictors and can often make predictions with an equal or better accuracy to the top clinicians, the top consultants, and do so in a fraction of the time. Mathematical ophthalmology is capable of making predictions. However, the accuracy of those predictions depend upon your having appropriately captured the underlying mechanisms. So they each have their strengths and weaknesses, and I say are complementary approaches. So as I said, I've coined this term, but I invite other people to use the term mathematical ophthalmology and similarly computational ophthalmology if they think it would be helpful. I guess there are sort of two reasons for that. One is it's helpful to have a name for a field, so you have something to refer to it by. But also, and importantly, I think in terms of trying to build a community and have a sense of identity as the people who work in this particular area. So if you want to refer to it in one catch-all, then you could use the acronym MACO, mathematical and computational ophthalmology. So getting on to the main topic of my talk today, I need to give you a little bit of background on the biology of the eye. So the diagram on the left here is of the eye viewed in the transverse plane. So if you were to cut the eye horizontally, you'd see something like this. This is a human eye. And the eye is basically a fluid-filled chamber enclosed by three layers of tissue. And these are the retina, which is the light-detecting layer, the choroid, which is a vascular layer which supplies oxygen and drains away other drains away other waste products and so on. And then the sclera, which is the white rubbery outer coating. The retina runs from the optic disc, where the optic nerve, retinal vein and artery puncture the eye, around to the ora serrata. It contains a number of different cell types, but perhaps the most key are the photoreceptors, which are your light detecting cells, and those come in two varieties, rods and cones. Rod photoreceptors provide low acuity, black and white peripheral vision under low light conditions, whereas cone photoreceptors provide high acuity central colour vision under well lit conditions. And these cells are quite heterogeneously distributed across the eye. So if you look at the figure here, this is showing the variation in rod and cone density with eccentricity. So 0 degrees eccentricity corresponds to the center of the eye, the retina, the fovea. And then as you increase in eccentricity, say nasally here, then you're moving around the eye over to the ora serrata. So you can see that cones are very densely packed in the center of the retina, in the fovea, and then they decrease quite sharply in density as you move out towards the periphery. Rods are absent from the very center of the fovea, from the foveola, and then they increase in density up to about 20 degrees eccentricity and then fall off more gradually in density towards the ora serrata. So the center of the retina is cone dominated, whereas the rest of the retina is rod dominated. If you were to have a look at the retina, this is a diagram that gives you a rough schematic of the different cell types. So here this is orientated in such a way that the bottom would lie innermost in the eye and the top would lie outermost, so light penetrates in from the bottom. The photoreceptors are in this outer layer here at the top, your rods and your cones, and those are but another layer of cells known as the RPE, or retinal pigment epithelium. If you zoom in on the photoreceptors, they look a bit like this. They have outer segments, which contain discs in which the photopigments are embedded. Inner segments, which contain the mitochondria, so the metabolically active parts of the cell bodies with the nuclei, and then axons ending in synaptic terminals. A key thing to note about these outer segments is that they're quite dynamic in that each day, with a rod, for example, you lose the top 10%, the tip of the outer segment sheds, and that's then phagocytosed or gobbled up by the overlying RPE cell, and new outer segment discs are generated where the outer segment meets the inner segment, and similarly for the cones. So the term retinitis pigmentosa, or RP, refers to a group of genetically mediated retinal degenerative diseases. So far, mutations have been discovered in over 80 genes, and this accounts for about 50 to 70% of cases. The majority of these are either autosomal dominant or autosomal recessive, though there are also X-linked and then in rare cases digenic and simplex cases. Ordinarily, it's non-syndromic, but there are also syndromic forms where other ailments are associated with it. So, for example, Usher's syndrome is the most common here, which is also involves hearing loss. Typically, people with RP, the genes that are mutant are expressed in the rods or the RPEs, but not in the cones. So if you're a patient with this condition, you'd usually experience night blindness in adolescence, and that it corresponds to the loss of rod function. As you move into early adulthood, this then develops into tunnel vision, which corresponds to beginning to lose cones, and then that vision progressively narrows over time until you, by about the age of 60 or so, you may lose central vision, although it varies quite a bit between patients. So it's a leading cause of blindness worldwide. About 2 million people globally have this condition. It's the most common inherited retinal disease, with a prevalence of about 1 in 3.5 to 1 in 4,000. And it's currently incurable in that we can't prevent a person from going blind or, say, restore vision completely once it's gone. There are, though, some treatments available to try and slow things down. These come in various varieties. One promising approach is gene therapy. So the first of these types of drugs, called Luxturna, was approved, I think, in about 2017. The great thing is that they can sort of sort things out at the source, but the downside is that it's specific to a particular mutation. So this one is specific to the RPE65 mutation. So you would have to develop one of these therapies for each mutation, and as I said, we only know 50 to 70% of the genes anyhow at this point. There are various other possibilities, including, for instance, apoptosis or neuroprotection, for example, treating with trophic factors or antioxidants. And if you do lose your vision, then you can get a partial restoration using artificial retinas. So the images on the right-hand side here are the fundi of a healthy and RP patient. So a fundus is the inner surface of the eye opposite the lens. It's what an optometrist sees with an ophthalmoscope when you go for your eye test. So the left one, as I said, is healthy, the right is RP, and the particular thing to notice here are these pigmentary deposits, which are known as bone spicules. Excuse me. And these occur when you've lost photoreceptors and hence vision in this region, and the RPE cells migrate into the inner retina and form sort of cuffs around the retinal vessels, and you get these pigmentary deposits. So there are two particular things I want to note about this condition. The first is that it's a rod cone dystrophy. So your rod photoreceptors degenerate because they express a mutant gene. However, the cones don't express this gene, which rather raises the question as to why they degenerate. So it seems that cones depend upon rods, either directly or indirectly. And various mechanisms have been proposed to explain secondary cone loss, and I'll describe those in just a moment. First, just the other thing I wanted to highlight is that there are quite characteristic and well-defined patterns of retinal degeneration, spatio-temporal patterns of retinal degeneration associated with RP. And these so degeneration tends to start in patches, which expand and coalesce. These patches tend to form in the middle far periphery, and then degeneration spreads peripherally and centrally. And these two sort of features highlighted in gold tend to be discussed separately in the literature, but I'd want to argue that actually they're intimately connected, and I'm going to explore that a bit in my modelling in a moment. So I mentioned that there are various mechanisms to explain secondary cone loss, and I'll just shoot through those quickly now. In each case, it starts the same way. You have a mutation, and that leads to the degeneration of rods. There are then 5 main mechanisms that have been proposed. The first is the oxygen toxicity hypothesis, which suggests that when you lose photoreceptors, that decreases oxygen demand, which leads to a rise in oxygen levels, and which then become toxic for the remaining photoreceptors, leading to further photoreceptor loss and hence further oxygen rise. So this is a sort of a self-reinforcing cycle. Second possibility is the trophic factor hypothesis. This suggests that rods produce a trophic factor that's necessary for cone survival, so that if you lose your rods, the trophic factor depletes and the cones degenerate. This is self-limiting because once the cones begin to be lost, then there's less competition for this trophic factor. Another possibility is that metabolic dysregulation, that as photoreceptors degenerate, the transfer of nutrients and waste products between the photoreceptors and the RPE and choroid is disrupted, leading to photoreceptor death. The toxic substance hypothesis suggests that when cells die, they release a toxic substance which kills their neighbours. And then finally, the microglia hypothesis is similar to the toxic substance hypothesis, but here, when cells die, that results in the activation of microglia in the inner retina, which then migrate into the outer retina, phagocytose or gobble up the rod photoreceptor debris, and then themselves release a toxic substance which kills neighbouring photoreceptors. So these are complementary mechanisms. It could be any or all of them in any combination. And two things to note about them is that all of them are self-reinforcing, with the exception of the trophic factor mechanism, which is self-limiting. And all of them, in fact, could explain both a portion of rod and cone degeneration, not simply cones, with, again, the exception of this trophic factor hypothesis, which would only explain secondary cone loss. So in today's talk, I'm going to focus in on the second hypothesis, trophic factor depletion. In the past, I've published work on oxygen toxicity, and I've got ongoing work in the other mechanisms, but it's this one I'm going to focus in today. So the other feature I highlighted was these patterns of retinal degeneration or visual field loss, and so that's what I'm describing here. So I'm going to relate this to different geographical regions in the retina. So here on the left, you see the centre of the retina is the fovea. If you move outward from that, you get to the parafovea, the perifovea, the mid-periphery, which contains the optic disc, and then the far periphery at the edge. So the diagram on the right then shows patterns of visual field loss, where black regions are scotomas or blind spots, and white regions are regions of preserved vision. And what I've done here is I've developed my own sort of visualization of a set of patterns that were described by Grover et al. Back in 1998. So they did a whole bunch of Goldman visual field tests on RP patients. These were longitudinal studies. They followed them over time and saw the spatio-temporal patterns of visual field loss that developed. And they categorized those into 3 groups and 6 subgroups. So Group 1 involved a restriction of the peripheral visual field, which was sometimes accompanied by a para or parafoveal ring scotoma. Group 2 had an early sort of superior visual field loss, which then wound around inferiorly, either nasally or temporally. And Group 3 had a mid-peripheral ring scotoma, which then extended either superiorly or inferiorly, winding around the far periphery. In each case, central vision was best preserved, although that eventually is also lost. So the overall approach that I'm taking in my research, and you'll see some of this in today's talk, is threefold. So you begin by isolating a particular disease mechanism, and this is a place where mathematical modelling really comes into its strength, because or one of its strengths, because you're able to isolate mechanisms using mathematical modelling in a way that you would never be able to do, or would be extremely difficult to do experimentally. So in this case, I'm going to be isolating the trophic factor mechanism. You can then use your mathematical model to predict the sort of patterns of disease progression that you would get, the patterns of visual field loss and hence retinal degeneration. And I should just note here that the patterns of retinal degeneration and the patterns of visual field loss are the same, except that you need to flip things vertically and horizontally given the pinhole effect of the pupil. And then lastly, having made predictions, you can then infer the strengths and weaknesses of your hypothesis. So if you pick out a particular mechanism and you find it is able to reproduce the patterns of degeneration that you see in patients, that suggests that that mechanism could indeed generate those patterns. It doesn't say that it does for sure, because it could be another mechanism, but it does show that at least it's capable of generating that pattern, or is consistent with it. Perhaps more interestingly is if a mechanism fails to generate a pattern, because then that reveals the necessity of other mechanisms being at play in order to give rise to that degeneration. So as I said, I'm going to focus in today on the trophic factor hypothesis, and this has a few strands of experimental evidence. Just before I do that, I'll just remind you how this hypothesis works. So you have a mutation that leads to rod degeneration. The rods are thought to produce a trophic factor which is necessary for cone survival, so that when the rods die, the trophic factor is depleted. That then leads to cone degeneration, but as the cones are lost, then the competition for the trophic factor decreases, so there's a sort of self-limiting step. So in terms of experimental evidence, well, this trophic factor has been identified. It's known as rod-derived cone viability factor, or RDCVF. It's known to be produced by rods, and it's been identified as a truncated thioredoxin-like protein. So this was identified by Thierry Levelard et al. Back in 2004. They've also worked out its mode of action. So it turns out that RDCVF is able to increase cone glucose uptake, and it does this by binding to a protein on the surface of the cones called, the cone membrane called basengen-1. That then binds to GLUT1, and then that increases the uptake by GLUT1 of glucose. And it's also been shown that RDCVF can slow cone degeneration and preserve cone function in chick, rat, and mouse models. So there's a strong case for this hypothesis, and indeed it's probably the hypothesis that's received the most attention from the experimental community over the last 20 years or so, although I think most of that has come from a particular lab, from Thierry Levelard's lab. So it's a strong contender as a possible mechanism in RP. So what I'm going to do is formulate a mathematical model, and then I'd like to pose a series of 4 questions to this model to see what kind of insight it can give. So the first feature I'm going to look at is, so as I mentioned before, you lose rod photoreceptors first in this condition, and that tends to occur in patches. So the first question is, if you lose a patch of rod photoreceptors, will a patch of cone loss follow? And if so, how wide will that patch of cone loss be in comparison to the patch of rod loss? The second question is, well, if I wanted to treat, say, by inputting RDCVF into the system in some way, what would the critical dosage be in order to prevent cone loss following rod loss? Thirdly, what sort of patterns, spatio-temporal patterns of cone degeneration, are explicable via this mechanism? And lastly, and this last question will become clearer when we get to it, what are the sufficient conditions to recapitulate in vivo patterns of cone degeneration? So I'll just go through these one at a time now. Before I do that, I'll just derive the model. So for the mathematicians in the audience, the model I'm going to derive is a reaction diffusion partial differential equation model in 1 dimension in spherical polar coordinates. If you're not familiar with partial differential equations, the way that these equations work is that they track the evolving density of cells and concentrations of chemicals in a particular region of space through that spatial domain and over time. So in this case, I'm going to determine spherical polar coordinates, and I'm looking at a polar angle between theta equals 0 and theta equals 1, where I've non-dimensionalized or scaled this angle to go between 0 and 1. I'm assuming symmetry about the z-axis in the azimuthal direction, and I'm effectively depth averaging in the r direction, so we're just reducing this to a 1D problem. Now, it's important in this model to incorporate and to account for the heterogeneous distribution of rods and cones. And to do this, I'm going to fit analytical functions to data from the literature. So this data comes kindly shared by Christine Curcio, who did some great work on measuring these distributions back in the early 90s. And so what I've done is come up with these analytical expressions and then I'm going to fit the B, capital and little b's, in order to get a good fit to that data. So the cone expression is basically a sum of two decaying exponentials, and the rod expression is a decaying, sort of modified decaying exponential with this theta at the front. So the top left panel here, you've got increasing eccentricity on the x-axis, so going from 0 degrees, which is at the fovea, around to the on highest eccentricity is at the ora serrata here. The black dotted line is the data, the red line is my approximation. This is for rod density. Similarly for cone density on the right. And then if you add those two panels together, you get the total photoreceptor distribution. And so again, there's a good match between the approximation and the data. And the reason for using this, coming up with this algebraic expression, is we need to do this in order to get that information into the equations. For simplicity, I'm not actually going to write the equations out as equations. I'm going to write them out as words, because I think, particularly for those who are not mathematicians in the audience, that will be clearer. But actually, you don't really need to see the equations to understand the talk, and the details are all in the publications anyhow. So there are 4 equations. The first is for the rate of change of trophic factor concentration through space and over time. And so this accounts for the diffusion of trophic factor either towards the ora serrata or towards the fovea, its production by rods, any input from treatment, uptake by cones, and any decay due to biological degradation. We then have an equation for the rate of change of the rod density, PR, and what we say is that that's either fixed or it decays exponentially due to the mutation that they express. And it decays at a rate phi r. And then a rate for the rate of change of cone density, and we say that they remain fixed in density unless and until the trophic factor concentration dips beneath a critical threshold F crit, at which point they decay exponentially. And then lastly, if we're considering a case with treatment, so I should say that photoreceptors, once they're dead, they can't regrow. So treatment couldn't restore lost photoreceptors. However, as I mentioned before, these outer segments of the photoreceptors are shed and renewed. And in fact, you can go down to a 0 length of outer segment and still, provided the rest of the photoreceptor is intact and sufficiently healthy, you can regrow the outer segment back to full length. So if we're looking at treatment cases, rather than looking at cone density, we look at cone outer segment length. And we say there that they'll shrink when the trophic factor concentration is below the critical threshold and are capable of regrowing when it's above that threshold. And then lastly, these are our governing equations. And then lastly, we need some initial and boundary data just to close the system. So in terms of initial conditions, I'll say either that everything's healthy initially, which is this panel here, or I remove rods from some interval, theta r1, theta r2, which is the panel on the right, where the blue curve here goes down to 0. This is the rod density between theta r1 and theta r2. And then lastly, boundary conditions, those just need to be applied for the trophic factor. And we say we have 0 flux boundary conditions, so they can't escape from either boundary. And that happens by symmetry at the fovea, because we're in spherical polar coordinates, and just as the simplest assumption at the ora serrata. So for clarity here, then our inputs to the model are theta r1, theta r2, the boundaries of the patch of rod loss, phi r, the rate of mutation-induced rod loss, and F crit, the critical thresholds for RDCVF, and the outputs are the evolving concentrations and densities of RDCVF, rods, cones, all the cone outer segment lengths. So the first question I'm going to look at then is, what is the spatial extent of cone loss consequent to a patch of rod loss? So to motivate this, I'm going to show a few sort of example simulations so you can see the sort of thing that happens. So what I'm doing here is I'm going to removing a patch of rods, and then I'm looking to see how the cone density evolves after that point. So in each case, these heat maps show eccentricity on the x-axis, time increasing on the y-axis, and the proportion of cones remaining compared to their healthy density is what's plotted. So if it's white, so if it's yellow, all of them are there, and if it's blue, they've gone. And I'm removing patches that I describe as wide, which have a width of about 0.1 when non-dimensionalized on the top row, and narrow ones which have a width of about 0.01 on the bottom row. And I remove them from left, central, and right positions here. So you can see that when the patch is wide, it gets this is the initial patch of rod loss is wide. The cones then degenerate in each case, and the width of cone loss is a little bit narrower than the width of rod loss. If we instead take out a narrow patch, in the central and right cases, you don't then lose any cones subsequent to rod loss, but you do in the left-hand case, again being somewhat narrower than the rod loss for the simulation. Well, wanted to sort of analyze this in a bit more detail, so I did a more detailed sort of sweep numerically, and also performed an asymptotic analysis to look at the leading order behavior of our equations, and this is what I'm showing here. So if you focus in on this panel I'm highlighting with my pointer, the second one from the left, this is an analytical solution, so I'm comparing with numerics, but focus on the analytical for now. The x-axis here is the position of the right-hand boundary of rod loss, and the y-axis is the position of the x-axis is the position of the left-hand boundary of rod loss, the y-axis is the position of the right-hand boundary of rod loss. And since the left-hand boundary is always to the left of the right-hand boundary, this is an upper triangular plot. And what I'm plotting in this heat map is the difference between the width of the patch of cone loss and the width of the patch of rod loss which generated it. And so if it's blue, that means the cone loss patch is narrower than the rod loss patch. And if it's red, that means that there are regions where the cone loss patch actually goes beyond, it is wider than the rod loss patch. Another way of visualizing this is to take sort of profiles through this heat map in a diagonal direction for patches of constant width, and that's what I'm showing in the panels on the right. So if you focus on the far right panel, then here each coloured line is showing a different patch width. The x-axis is the position of the middle of the patch of rod loss, and the y-axis is showing you the difference between the width of the patch of cone loss and the width of the patch of rod loss. So you can see that the patch of cone loss gets wider compared to the patch of rod loss as you move towards the fovea, towards the centre, and also that close to the centre there are regions, as I say, where the patch of cone loss is wider than the patch of rod loss, and that happens more often for narrower patches of rod loss. So that answers the first question. The second question we wanted to look at was in terms of the critical treatment dosage required to prevent cone loss once you've lost a patch of rods. So again, I'll show a sort of motivating situation scenario. So suppose that I remove a patch of rods from some interval between 0.3 and 0.7 here. So this is increasing eccentricity again on the x-axis, time increasing upwards. The RDCVF concentration, therefore, in that region is going to decrease, so it's a deeper shade of blue in this region. And the cone outer segments, notice its outer segments we're plotting now, are going to degenerate. If then I treat across the whole retina with RDCVF, then the RDCVF quantity will increase across the retina, as you see here. That's not going to affect the rods because they're not dependent on the RVCVF, but the effect is to restore the cones in that region. So the question that I was interested in here then was, well, what would the critical dosage be, and how will that depend upon various features of the model? And there are two broad kinds of treatment you might think of applying here. The first is what I call a global treatment, where the whole retina sees the treatment. So that could be because you've injected intravitreally with RDCVF or a retinal gene therapy, so it goes into the jelly in the centre of the eye and the whole retina sees it. Or alternatively, you could use something like an encapsulated cell-based delivery, where you put a little capsule in which contain the cells which produce RDCVF, providing a sustained delivery via the vitreous to the retina. Or you could imagine a local treatment, which was maybe a subretinal injection just beneath the patch of rod loss, so local to that region, again either with RDCVF or retinal gene therapy. So these are the two scenarios we want to consider. So let's focus first on this left-hand diagram. So I'm plotting here the critical treatment dosage needed, assuming it's a sustained dosage, in order to prevent cone loss following a patch of rod loss. The x-axis here is the left-hand boundary of the patch of rod loss. So here, solid lines represent global treatment, dashed lines local treatment. So in the left-hand boundary for a wide patch of rod loss, you'll see that the solid and dashed lines overlap each other, which means that the critical global and local treatment concentrations are the same, or rates are the same. Also notice that the treatment rate is higher as you move towards the fovea, towards the centre. By comparison, if you look at a narrow patch of rod loss, we still have the trend that the critical treatment rate is higher towards the fovea, but now we can distinguish between the dotted and solid lines. So what we find is that for a narrow patch of rod loss, the critical dosage is higher if you treat locally than if you treat globally, and that this becomes further exaggerated as you move towards the fovea. So the third question I wanted to look at was which patterns of cone degeneration are explicable via this mechanism. So to look at this, let's focus on this bottom panel. And so what I do here is I assume we start with healthy initial conditions, and then we allow rods to degenerate exponentially across the domain which is driven, we're assuming here, by a mutation. And then we look to see how the cone density evolves over time as a result of that. So here I'm plotting the sort of the proportion of cones that remain compared to their normal healthy densities at any location. So yellow means they're all healthy, blue means they've been lost. And again, eccentricity is increasing from the fovea to the ora serrata along the x-axis. Time is increasing vertically. So what we see is that degeneration starts at the centre of the retina and propagates outwards towards the periphery. So this is actually quite surprising because this is, well, firstly note that this is completely the opposite of the patterns that you see in humans, right? So in RP, degeneration starts at the periphery and it moves inwards towards the centre, and the pattern that we're seeing here starts at the centre and it moves outwards towards the periphery. And I guess the surprising thing here is that this mechanism has received probably the most attention, as I said before, in the experimental literature in the last 20 years or so, and yet actually it doesn't seem to be able, at least under the simplest of assumptions, to generate the patterns you see in patients. So in some ways it's surprising, in some ways it's not surprising if you consider the ratio of rods to cones through the eye. So this is showing rod-cone ratio, and you can see it's a monotone increasing function. So there are far more cones per rod at the, or far more rods per cone, far more cones than rods, cones per rods at the fovea than there are at the ora serrata. So you would expect then cones to become starved of RDCVF first at the centre. So in some ways it's surprising, in some ways it's not. But this led me then to the sort of final question, which is what might sufficient conditions be to recapitulate these in vivo patterns of cone degeneration? I was dealing with the simplest of assumptions previously, because we modify the assumptions of the model to find a situation where we could reproduce these patterns, which seems particularly worthwhile goal, given that, as I said, this mechanism has received an awful lot of attention and experimental validation in the literature. So of all the parameters in the, up until this point, all the parameters in the model have been assumed to be spatially uniform, not to vary in space. Of all the parameters in the model, only two of them could realistically vary through space, and those are the rate of mutation-induced rod loss, or FIRE, which is known to vary spatially, and the RDCVF threshold concentration, FCRIT, which again we would expect to vary spatially. So the question is, can we find profiles for these, spatially varying profiles that would allow us to reproduce the patterns we see in vivo? So in order to answer that question, we're going to formulate something that mathematicians call an inverse problem. I do that both numerically and analytically. And so the basic idea then is that whereas before we started with some parameters as our input and then asked what pattern of cone degeneration we would get as our output, we're now reversing this process and saying, well, given a known pattern of cone degeneration, what does that tell us about what the profile of FIRE or FCRIT had to be in order to generate that pattern? And I just note here that in any given simulation, what I will do is either keep vary FIRE spatially but assume that FCRIT is spatially uniform, or vary FCRIT spatially and assume that FIRE is spatially uniform. I won't vary them both at once. So in order for this to work, I need to know what pattern I'm expecting to get. And so I won't show all of these, but let's just focus on the bottom right panel here. So this is pattern 3, where you have an early loss, as you see in this little inset, of the mid-peripheral retina, and then that spreads inwards and outwards. So what I'm plotting here is eccentricity on the x-axis and time of degeneration on the y-axis. And so time needs to be smallest or earliest here in the mid-peripheral region. And then as time marches forward, we want degeneration to spread towards the periphery and again towards the centre. And there's multiple sort of various qualitatively different forms that I was using of this sort of shape, but the different forms don't matter. The main thing is the overall shape. So then, given that target, and I'm just going to keep sticking with pattern 3 here, what would these inverse solutions be? So the left-hand column here shows you what how FIRE, the rate of mutation-induced rod loss, should vary across the domain, and the right-hand column shows you how FCRIT, the critical RDCVF threshold, should vary. Don't worry about the rows; they're just for different functions. So just focus on the top row. But you see in both cases that these functions need to be at their highest in the region where we want degeneration to start. So that makes intuitive sense in that in those locations where the mutation-induced rate of rod degeneration is highest, rods will be lost first, and hence trophic factor depleted first, and hence cones will degenerate first. And similarly, if you have a region where the trophic factor threshold is higher, you're going to pass beneath that threshold sooner. So it makes sense that they would have these sorts of shapes. So to summarize then, in answer to our first question, we found that patches of cone loss will follow patches of rod loss, and that those patches of cone loss tend to be wider towards the fovea and in some cases can exceed the width of the patch of rod loss. We've been able to calculate critical treatment rates using RDCVF. We've shown that those need to be higher for more central patches of rod loss, and that where you've got a narrow patch of rod loss, the local dose concentration should be higher than a global dose concentration. So under the trophic factor hypothesis, we're not able, under the simplest of assumptions, to reproduce in vivo patterns of degeneration, which in some ways is surprising given the level of attention that the mechanism has received. However, we are then able to modify the model to consider spatially varying profiles for the rate of mutation-induced rod loss or the trophic factor threshold, which are required to recapitulate those in vivo patterns of cone degeneration. So in theory, these are experimentally testable predictions that someone could go out and test and see if indeed these criteria are satisfied in order for these patterns to be generated via this mechanism. Phi-R has been measured in the past, although not quite in the detail that we need for this, but that could easily be measured. Well, I say easily, I'm not the one who'd have to measure it. FCRIT would be a bit more challenging, but I think is theoretically possible, but that would need a bit more thought. Just in terms of how I see this work going in the future. So I've been talking in the past. I've done work in the oxygen toxicity hypothesis, and these are the sorts of patterns that we see being generated. Today we've looked at the trophic factor hypothesis, which generates this kind of pattern, and sort of ongoing and future work. I'm expecting from the metabolic dysregulation and toxic substance hypotheses these sorts of patterns to come. So you can think about how those sorts of mechanisms might combine together to generate the full patterns that we see in patients. For example, pattern 1 could be a combination of oxygen toxicity and trophic factor depletion. Pattern 2 could involve all 4 mechanisms, and pattern 3 again a combination of them. Now you could imagine maybe you could combine these mechanisms together in different ways to generate these patterns, but the important thing is if you're a clinician and you want to try and treat this disease and slow the rate of progression, then it would seem from this modelling that the disease is divided into different temporal stages where different mechanisms will drive degeneration, and that also different spatial locations in the retina may be more susceptible to some mechanisms than others. So that would then have implications for how you treat at different temporal stages in the disease and at different locations in the retina. Before closing, I just wanted to highlight a couple of things that might be of interest to people if they want to learn a bit more about mathematical and computational ophthalmology. One is I've created and have been guest editing a special issue in the journal Investigative Ophthalmology and Visual Sciences in MACO. Submissions are now closed, but do take a look if you're at this link if you're interested to learn a bit more. There are 20 papers so far out with others to follow, and this includes invited reviews from the following authors. 2 of those have been published already, the rest will follow. I also have my own seminar series in mathematical and computational ophthalmology, which are live online, and then recordings are maintained on YouTube. So these are the talks that we've had so far, and the next talk will be on Monday, the 19th of August at 3 p.m. British Summer Time from Christine Kersey on why AMD is a mathematically tractable disease, and the link's just here for that. And then lastly, this has happened now, but I organised a special interest group with the Association for Research in Vision and Ophthalmology, again in mathematical and computational ophthalmology. So this was had talks from a few different top speakers, and also a discussion around current opportunities and challenges in this field. And so the link to that recording is there. I think if you're a member of ARVO, you can get it for free. If you're not, you'd probably have to pay a small fee. And I'm also looking to form, I wanted to highlight a subgroup within the Society for Mathematical Biology in mathematical and computational ophthalmology. To do that, I need to form a petition, and the people whose names are on the petition need to be members of SMB. So if you are a member or would be happy to join and be interested in that subgroup, then please do let me know, and I'll add your name to the petition. And then, sorry, just one final thing is that I wanted to highlight that if you are an early or mid-career scientist, or you know early or mid-career scientists, you might be interested in this paper that I wrote called Advice to a Young Mathematical Biologist, which provides career advice to early to mid-career researchers, not just in mathematical biology, but across the life, experimental, clinical, physical, mathematical sciences. The advice doesn't come from me, it comes from 10 current and former presidents of the Society for Mathematical Biology, and covers topics like finding and working with a mentor, applying for funding, giving your first lecture, and all those sorts of things. So the link to that is just down here at the bottom. So just in this final slide then, I'll just say I've been talking to you about inherited retinal degenerations today, and I've got continuing work in that area. I've also worked and continue to work in retinal metabolism and oxygen distribution, other diseases of the eye like AMD, and also visual neuroscience, and also in areas of mathematical biology outside of that. If you'd like to learn more about my research, do just take a look at the Twitter feed or my website down there. So thanks ever so much for listening, and I'd be happy to take any questions that you might have.
All right, thank you, Paul. So the floor is now open for questions. Feel free to just unmute and ask if you have any.
Hi, Paul.
Hi, Joel. Good to see you.
Good to see you. Good to see you. Yeah, very nice talk. Thank you so much for your talk.
Thanks.
You know, I'm also working with the eye, as you probably remember, and it's interesting to see more people starting to show a little bit more interest in what the models can do for them, right? Clinicians and other people that are not so close to what a model can do. But one thing that I listen over and over again is how we can make the models more biologically meaningful, right? And that is a very hard thing for us to solve. So my question is a little bit towards that to you, like, how do you solve that problem of including more biology in your model, but not to a point that is in a hindrance to your model, but it's actually meaningful, that is something that we can validate and create more confidence in these models?
Thanks. That's a great question. So, well, I think as a sort of working pattern to start off with, I think it's really important to be working in close collaboration wherever possible with clinicians or experimentalists. I think there's no substitute for being able to have regular conversations with someone, both as you try and identify the problem you want to work on, as you're developing the model and interpreting the results. I think throughout that process it's important to be having that back and forth. That makes it often a more time-consuming process, but I think it's worth it because the model at the end is much more valuable. Of course, it's important to familiarise yourself as much as possible with the literature, but there is something to be gained from having that actual relationship with a living, breathing experimentalist that you can't get just from reading papers. I guess the question of like how much biology do we include in models is sort of a bit of an age-old question with mathematical modelling. You want to put enough detail in to capture the essential features, but not too much that you end up with something that's more complicated than you needed, or that it could end up being as complicated as the thing that you're starting off with. The point of modelling is that you're trying to come up with something that's simpler, a simplified representation of reality, so that you can make some kind of progress and gain some kind of insight. I guess it's kind of has to be taken on a case by case basis. In the work that I was presenting today, I try and isolate just one mechanism and ask a this sort of what if kind of question as to what would happen if that was the only thing that was happening. I think if you try and throw everything, including the kitchen sink, at it, then it's hard to see the wood for the trees, and you don't know which mechanism is important. So I think that partly just comes from experience, and just getting better with modelling as you get it. It's not something you can sort of like teach quickly in an undergraduate degree, or something you need to learn it through a PhD and subsequent research. So I guess the other thing is trying to communicate the value of what you're doing and the meaning of it to biologists. You might have come up with a great model, but if you don't then communicate that well, that its impact could be decreased. So part of that is if you're able to, it's nice. I mean, I think it's good to publish in a variety of journals, but so I try and publish work in the more mathsy journals, mathematical biology journals, but it's also good to try and get a footing in more clinical or experimental journals. So, for example, with my special issue in the IOVS, I think that's a good way of raising the profile and the possibilities and value of these kinds of approaches. I think the other part of that is just it's difficult, but trying to write papers in such a way, or communicate and give talks or attend conferences, and try and communicate in a way where you don't blind people with the mathematics, but you just try and explain to them what the biological significance is. I think, though, in general, there's a sort of, I think you were sort of hinting at this, there is an increasing openness and appreciation amongst clinicians and experimentalists as to the value of a mathematical approach. And indeed, actually, ophthalmology in many ways is leading the way in this regard, in that on the computational side, in terms of artificial intelligence, not just the first, but the first two artificial intelligence algorithms to be used in a clinical setting, both in ophthalmology. They're both to detect or diagnose people with diabetic retinopathy. So I think the first, at least, of those was approved for FDA use by the FDA in 2017. So it's quite it's also we're blessed actually in ophthalmology with a vast amount of data, particularly with things like OCT having been around for 20 so years or now. I think I heard a statistic a while ago that this was a few years ago now that one OCT scan is taken every 3 seconds on planet Earth. So there's a vast quantity of data there to play with. So I think we're in a good position to make real inroads, and that there's a great there's a great value in this quantitative approach, and as I said, an increasing awareness of that value. So exciting times for mathematical and computational ophthalmology, I think. Sorry, that was a rather long, rambly answer.
No, it was a great answer. Yeah, thank you so much. It is very difficult, right? I think it depends very much on the hypothesis you want to test, right? If you have the hypothesis, you need all the pieces that those hypotheses are at least based on, right?
Yes,
it's basically what you said. Yeah. There was another thing that I wanted to mention, since you talked about the number of data you have. I found that now there is even a camera that they send to you so you can attach on your phone, and the person can take their own scan. So that makes things like having these time series very much easy for us, right? That is modeling. Do you see, like, have the opportunity to see the type of data that can generate, you can gather with that? Like, is it reliable? That's basically my question, right? Because it's a user doing it. It's not a doctor or nobody that was trained.
I haven't. I've walked past tables at conferences where people have had these devices, but I haven't actually spoken to the individuals who are selling them. So I can't say for sure how reliable they are. My guess would be that they're pretty good, but it might depend a little bit on how careful the user is and how they apply it and what the conditions are. But I think there does seem to be a move in general, including things like wearable technologies of the ability to gather data, for people to gather their own data effectively, and that seems to be a sort of exciting development. So I think probably I would imagine the image quality. I don't know, so I can't say for sure. But I'd imagine the image quality or the consistency of that would probably not, I would guess, would not be as good as if you went to an optician's or an ophthalmologist. But that is probably made up for by the fact that you could have rather more frequent images or a larger volume of data. So I think there's definitely value in it, and it's exciting that's now a possibility. I guess the other thing is that there's a limit to the kinds of scans you could do in that way, right? So you're getting a photograph of the back of the eye, but you're not going to be able to do an OCT scan with your smartphone. That's still, maybe they'll be able to do it one day, but I think we're a long way off from that kind of. So the data would be more superficial, I guess.
Right. Yeah, I have other questions, but I want to let the opportunity for other people too. Yeah, if not, did you use any animal study? Any animal study helped you during the development of the model, or you just worked with humans? And if so, like, how the translation between the animal? We know that there are some differences in the eyes between these species.
Yeah, so in this case, I was mainly interested in the human eye. And so, for example, the photoreceptor topography data that I used was from a human eye gathered by Christine Curcio. And indeed, if you're interested in the human eye, then animal models of RP are less useful in general, because animals have eyes differ in various ways. In particular, most of them don't have this sort of special macular region and this particular distribution of rods and cones or anything quite so heterogeneous. So there are other animals with foveated retinas, like birds of prey and macaque monkeys, that kind of thing. But that's different. So there is alternative data there that's useful. You could take this model, put in those other photoreceptor distributions, and run it and see if you can reproduce the patterns you see in an animal. And that's something I'm planning to do in the future. But if you're interested in humans, then there aren't that many animal species that are sufficiently similar that it would be so useful. So I guess mostly the data from this is from is human data. I guess there might be a few measurements that would have been made in animals, like, so for instance, not in the stuff I did today, but in the past, I need to know, for example, did the diffusivity of oxygen in the retina, that was measured in an animal, but you would expect that to be the same as in a human. So there are a few sort of parameters that can come from animals, but wherever possible, I get it from human data.
Thank you. Thank you so much again.
Thank you.
Yeah, so I was kind of interested in what you were saying about one of the benefits of mathematical and computational ophthalmology being that you can sort of isolate different mechanisms and test to see, what the behavior is of just that mechanism. And I was thought that it was kind of compelling that it seems like with your current model that you presented today, it's kind of actually progresses in the opposite way than we see in patients. I was wondering maybe if you could say, and then you talked a little bit about kind of reverse engineering the starting parameters maybe that you would need to mimic that behavior. I was wondering if you could say a little bit more about that. So it's like, does that kind of the opposite behavior indicate that it's probably not this mechanism alone that's causing the vision loss we see in patients, or does the model need more retooling to kind of mimic what we see?
So, I mean, as I said, if we go with the simplest of assumptions, which means that all the parameters are spatially uniform, we do get the reverse pattern. So if this mechanism is to generate those patterns, we do need to do this sort of reverse tooling, as you said it. So we do need to conduct the inverse problem, and basically the one of or both of the rate of mutation-induced rod loss or the trophic factor critical threshold would have to vary spatially. The other sort of factors in the model that I consider I can't see any good reason why they would vary spatially. So those are basically the two options. And there are good reason to suppose that those would vary spatially. So we know from measurements in animals and humans that the rate of mutation-induced rod loss is not uniform across the eye. It does vary. So there's good grounds for supposing that would vary. The trophic factor concentration or the critical threshold across the eye would be a much more difficult thing to measure, partly because the threshold is a tad abstract anyhow as a notion. But that it should vary seems pretty likely. So, for example, I didn't show a plot in the presentation, but given that the ratio of rods to cones is this monotone increasing function, that you've got loads of cones and barely any rods, or no rods at the very center, and loads of rods but not very many cones at the periphery. Even under healthy conditions, right, the trophic factor concentration would be vastly smaller at the fovea than at the periphery, which suggests that somehow the eye is able to manage in the fovea with having a much lower concentration of RDCVF. So there must be something that's protecting it so that it can make do without having so much. So there seems to be, so that would suggest indeed that this threshold must be lower at the fovea than it would be at the ora serrata. Whether that's because other trophic factors are present and there's a spatial gradient of other trophic factors that are doing that protection, or it's something else, is not clear. I know at least from animal studies, I can't remember if there's human studies as well, that there are trophic factors that are known to have a spatial gradient across the eye. So there's good precedent, A, for having a spatial gradient of, say, maybe a threshold in this one, and also for having added protection at the center. Yeah, so I've thrown down the gauntlet. Here are these two things. Go and measure them. If it's not satisfied, then I don't think it's terribly likely that this mechanism is responsible for those patterns. It still wouldn't mean that it's not happening. I mean, it's clear they've identified the method. This RDCVF, it is there. It is playing a role in helping cones to absorb glucose, but it may not actually be so important in generating as far as this pattern of loss goes. It may be these other mechanisms that are responsible for the pattern, even if it's sort of there in the warp and woof of things. Yeah, so I await someone to go and experimentally verify or dismiss these predictions.
Gotcha. Great. Any last questions before we break? All right. Well, if there's no more questions, then I will extend a thank you to Paul for coming and giving a great talk today.
Thanks for having me.
Yeah, and we
Thank
will
you. Thanks.
go ahead and break for the day then.